Chapter 7, Protozoa. Most gardeners first poked and prodded protozoa as part of a biology lab assignment, which invariably involved identifying and sketching cell parts of a paramecium. They may therefore remember that protozoa are single-celled organisms with a nucleus, which makes them eukaryotic and therefore, along with fungi, members of the domain Eukarya. Protozoa, which term we use descriptively in this book as shorthand for a group of non-algal, non-fungal, animal-like unicellular organisms across several kingdoms, but don't get us started, are almost always heterotrophs, meaning they cannot make their own food. Instead, they obtain their nutrients by ingesting bacteria primarily, but also the occasional fungus and to a lesser extent other protozoa. Paramecium are still the favorite microbe. That's because these and other soil protozoa are considerably larger than bacteria, 5 to 500 micrometers versus 1 to 4 micrometers. They may still seem small to you, but in the scheme of microorganisms, 500 micrometers is pretty large. So large that under ideal lighting conditions, a paramecium, at least in water, is visible to the human eye. You still have to look very carefully and certainly will not be able to differentiate any of those internal or external features you were told to label in school, but without a microscope, you can see them flitting around. Through an electron microscope, unseen detail is observable. Protozoa are something to stay away from if you are as small as a bacterium. By way of comparison, if a single bacterium was the size of a pea, a paramecium would be as large as a watermelon. This is why bacteria can hide from most protozoa in soil pores that are too small for the protozoa to reach into. Another way to make the comparison is to go back to that same teaspoon of good soil with its billion-count bacteria and only several thousand protozoa. Over 60,000 kinds of protozoa are known, and contrary to any residual youthful hope you may have that they only live in pond water, a majority of them live in the soil. However, all do require moisture to lead an active life. Given the crucial role protozoa play, a quick review of some school biology, and then some, is in order. Amoeba, ciliates, and flagellates. Protozoa come in 3 basic models. First are the pseudopods, single-celled animals with amorphous forms most will remember as amoeba. These are constantly on the move, a feat, if you will pardon the poor pun, accomplished by pouring their cytoplasm, the soup with all its life parts, into one or more false appendages called pseudopodia, false feet. Pseudopods themselves are of 2 types. The first has a shell-like exoskeleton and 5 predefined holes. Think of a bowling or a golf club, through which the pseudopodia can appear. The other class lacks any shell or predefined pseudopodia. These amoeba are relatively large microorganisms, and many would be as visible as paramecium if they weren't so transparent. Amoeba lack a mouth and ingest bacteria by surrounding them and engulfing them in gas bubbles, into which are transmitted digestive enzymes. The entire vesicle is then absorbed and waste products subsequently expelled. Next in size are the ciliates. These protozoa are considerably smaller than their amoeboid cousins, but still much larger than their bacterial prey. Ciliates are covered with rows of hairs that beat like the slaves' oars on a Roman galley, propelling the organism to food or away from enemies. In addition, these oars create currents that bring bacteria into the ciliate's mouth region so they can be ingested. The familiar paramecium is a ciliate protozoan. The third and smallest type of protozoa are the flagellates. Their one or two long whip-like hairs, or flagella, allow them to move about in search of food. A few flagellates, like Euglena, the classic freshwater flagellates of pond water, produce their own food via photosynthesis and are thus autotrophs. Most, however, are heterotrophic, obtaining nutrients from eating and digesting other organisms in the soil. More symbiotic relationships. As so many of the soil food web organisms do, protozoa form symbiotic relationships, particularly with bacteria, to such an extent that such associations appear to be the norm rather than the exception. A classic example is that of the flagellates residing in the guts of termites, which digest the wood fibers the termite eats. We now know that the relationship is actually a three-way one. Electron microscopy reveals working microbes in the gut of the termite as well. These fix nitrogen from the atmosphere for the flagellates. Not often do you find a triple symbiotic relationship, though surely more will be discovered as exploration courtesy of the electron microscope continues. Lots of ciliates, too, enter into symbiotic relationships with bacteria. Some ciliates live in sand and farm bacteria colonies, and it is the methane-generating archaea inside ciliates that are responsible in part for the methane gas that develops in some ciliates as anaerobic respiration takes place. Population control police. Protozoa are attracted to and enter into an area where there is a good supply of bacteria in a pretty consistent progression. On average, a protozoan can eat 10,000 bacteria a day. First come the flagellates, the smallest of these microbes. These can move into small spaces in the soil, places where the large protozoa cannot, and where bacteria are plentiful. Even after the larger ciliates arrive on the scene, the still large population of bacteria provides plenty of sustenance for both the original flagellates and the newer ciliates. Finally, amoebae move through in search of bacterial prey, and also smaller protozoa. The combined pressure on the bacterial population becomes so great, numbers start to diminish. As readily available bacteria become harder to find, the larger ciliates and amoebae start to eat more of the smaller ciliates and flagellates. This reduces the population of ciliates and flagellates, which, in turn, allows the populations of bacteria to stabilize and return to a level that maintains the soil food web balance. Why aren't all the bacteria consumed by protozoa? One reason is that protozoa are restricted by bacterial slime. This film is hard for them to penetrate, and it lacks the oxygen that they require. Another reason is that the bacteria are smaller and able to hide in tiny soil pores. It seems counterintuitive that increasing protozoa populations most often results in increases in the bacterial populations upon which they prey. This occurs because fewer bacteria means less competition for nutrients among the surviving bacteria. Not having to compete all the time for food means they can divide well-fed. Likewise, their progeny will have something to eat so they can multiply as well. If protozoa can keep their own numbers in check, they have all the bacteria and fungi they need to eat. It is not just populations of bacteria that protozoa keep in balance. In their search for sustenance, some protozoa attack nematodes. Others reduce nematode populations by competing for the same limited food resources, i.e., other protozoa and fungi. This also helps keep bad guy nematode populations from flourishing. Protozoa need moisture to live, travel, and reproduce, and hygroscopic water, that thin film of water left on the surfaces of soil particles and aggregates, provides it, under normal soil conditions. If things dry up, however, most protozoa stop feeding and dividing and go dormant, encasing themselves in a cyst. How long protozoa can survive in this state varies from species to species. Some can withstand an extended dry spell of several years. This technique ensures the survival of both the protozoa and the plants that benefit from the nitrogen and other nutrients released by their activity. Mineralizers. Of critical importance to the workings of the soil food web are the waste products produced when protozoa ingest bacteria or fungi. These wastes contain carbon and other nutritional compounds that had been immobilized but are now once again mineralized and made available to plants. Nitrogen compounds, including ammonium, NH4 positive, are among them. If nitrogen-fixing bacteria are present, remember these usually require a pH of 7 or above to have good populations, free ammonium is converted into nitrates. If not, the nitrogen remains in ammonium form. Mineralization of nutrients is crucial to the survival of plants in a natural system. Our premise is that by interfering with or destroying the soil food web, the gardener has to step in and do extra work, making gardening a chore instead of an enjoyable hobby. If you are not convinced, then consider that as much as 80% of the nitrogen a plant needs comes from the waste produced by bacteria and fungi-eating protozoa. Since bacteria and fungi are attracted by plant exudates to the rhizosphere, and that is where protozoa consumes them, a huge source of plant food is delivered right around the roots. Other soil food web functions. All protozoa participate to some degree in the decay process by inadvertently ingesting small particles of organic matter. These are then broken up into smaller pieces, if not totally digested, and become available to bacteria and fungi in the waste stream. And other soil food web members rely on protozoa as one of their food sources, another reminder that it is a soil food web, not chain, with which we are dealing. Certain nematodes, for example, are dependent on protozoa as their food source and have developed specialized mouth parts to better ingest them. Worms, too, rely on healthy populations of protozoa. Without protozoa in the area, gardens are devoid of worms. Similarly, many microarthropods require a healthy dose of protozoa to thrive. Finally, not all protozoa are beneficial. Several kinds eat roots, but in a healthy food web, these are kept in check by other cannibalistic protozoa. So to a certain degree, protozoa serve as a food source for themselves and remain, even the worst of them, crucial characters in a healthy soil food web.